Okay, it's recording. Not sure how to present. Okay.
Okay, so our group is Raylan, Genevieve, Diana, Jeanette, and myself. We're doing reproductive technology, human-animal hybrid transplants. So the ethics of creating human and animal hybrids for organ donation has been a challenging topic for many. There's many point of views on it. What it is human-animal hybrids are being designed as organ incubators, whereby human stem cells are inserted into the embryo of an animal, with the animal acting as a surrogate for the human organ. Organ transplantation is the most promising curation for end-stage organ disease. However, the donor organ shortage has become a global problem that has limited the development of organ transplantation. And this is why scientists are looking for other sources on how to help people with end-stage organ disease. There have been some experiments done, human pig, human sheep, human monkey, that we will share and go over. There are some regulations. They're trying to figure out how to best go about this as a nation and in the world in general, worldwide. So as of now, it seems that China has less regulations than the US, with the WHO implementing a global registry for human genome editing to develop global standards for governance. It is unclear at this time, though, how this all may fit together within the policy. Done.
So between the 17th and 20th centuries, scientists transfused blood from varying animals into patients with different pathological conditions. Results were mixed, and most were likely unsuccessful because of the infectious agents in blood with animal to human blood transfusions. In the 19th century, skin grafts were also carried out from a variety of different animals, including sheep, rabbits, dogs, cats, chickens, and pigeons, but frogs were the most popular. Some of these grafts were successful when used to cover skin ulcers that allowed the tissues to grow underneath, and they used these for grafts. In the 20th century, it heated the way for more complicated scientific efforts involving like surgical techniques. So from 1963 to 1964, a man named Keith Reemsta transplanted chimpanzee kidneys into 13 different patients. Most of the transplants failed within 4 to 8 weeks. Although there is one patient that lived for 9 months after transplantation until ultimately dying from electrolyte imbalances. In 1964, another surgeon named James Hardy, he attempted to do a heart transplant from a chimpanzee to a semi-comatose human. Ultimately, the chimpanzee's heart was not large enough to support the circulation though, and it failed within 2 hours. Another surgeon named Tom Starzl also attempted to put a chimpanzee liver into a human in the 1960s without success. But in the 1990s, due to the improved immunosuppression therapies that they had created, one patient did survive for 70 days after a transplantation of a chimpanzee liver. There's also a story of baby Faye in 1983. It is one of the most known clinical cardiac cross-species transplants. Her heart was failing from a congenital defect, and a surgeon named Leonard Bailey attempted to transplant a baboon's heart into the young girl. And the surgical procedure was technically called successful, but unfortunately, due to the ABO incompatibility, baby Faye did die 20 days later. You can go to the next slide. Human-animal hybrids are currently being designed to act as organ incubators. The ultimate goal is to have an animal act like a surrogate for human organs and then be used for transplantation processes later on. So in 2017, Juan Balmente and his international team developed a first embryo containing humans and pigs. This utilized the human pluripotent stem cells injected into both pig blastocytes and embryos in the surrogate pigs. The efficiency of this success was ultimately low, and the implanted embryos only survived 20 to 28 days and showed signs of developmental retardation. In 2018, Pablo Ross from the University of California successfully created human sheep hybrids as well. They used these to increase the levels of human cells with the sheep embryo by using lead, and only 1 in 10,000 cells were said to be human, and this level was too low to be considered transplant ready. And one of the most successful and recent studies, again, Juan Belmonte developed a successful hybrid of human monkey embryos. The scientists disrupted the regulatory genes needed for various organs and got rid of the potential defects by injecting the embryos with replacement human stem cells from monkeys. The embryos were still alive out of 100, or 3 embryos were still alive out of the 132. And by the end of the study, they were ultimately destroyed after spending 4 weeks inside the surrogate mother. Let me go to the next slide.
Still muted. Can you guys hear me?
Yes.
Okay, so there are some successes that have created like new pathways that we can consider for human animal transplantation. There is a study that has engineered pig embryos that lacked skeletal muscles by deleting the genes that created the skeletal muscles using CRISPR-Cas9. And then they used the somatic cell transfer method to deliver the human-induced pluripotent cells or the porcine blastomeres from the control to supplement the lacking muscles to see if like they would be viable. And as a result, they actually rescued the embryos that didn't have these genes and were able to successfully produce viable and functional intraspecies chimeras. They were actually delivered as piglets, which show proof that we could potentially use some of the gene editing that can help make animal to human transplant a little more successful, or at least with getting some of the organs. The second study involves a post-cardiotomy ischemia reperfusion injury in pediatrics that needed extracorporeal membrane oxygenation and under cardiogenic shock are treated with mitochondrial transplantation. So in this study, the healthy mitochondria injected into the children were proven to reduce injury and damage to the healthy cells. And functional recovery after the ECMO removal was shorter in the experimental group and no effects on white blood cells and lactates, in addition to improved ventricular function. This could help with improving the success rate of animal human transplantation in future studies by reducing the occurrences of the ischemia reperfusion injury, which are common in a lot of organ transplantation procedures. Next, please.
So some of the ethical concerns arising with the human animal transplantation, there are several, being that it is a new and upcoming research more and more. One of them being animal welfare. So the animal cruelty, this goes with discussing between medicinal purposes and consumption. So a lot of people are wondering of the ethical concerns of the animals. As some research also already has pointed out that it's no different from raising animals for medicinal purposes compared to consumption purposes. Another ethical concern is the transfer of retroviruses. This poses a threat because animals' genomes can be transferred into humans. Animals are indeed susceptible to these viruses, and fear is growing based on the possibility of human tissues and animals being able to cause new zoonoses. So just a new species with the two of them combined, and thereby threatening the safety of human subjects in research for research purposes. So that is just the concern of safety versus risks, and how great is one or the other to continue forward with research in this area. Last one is a crossing of species. So a crossing of species raises ethical concerns, as humans and animals are different entities and have different rights. For example, animals have no rights like humans do, as far as they're not legal entities when it comes to research purposes. And other ethical concerns regarding the crossing of species include the human-like consciousness. If human stem cells are placed into animal embryos, the question rises, will they have human-like consciousness? Will the human stem cells transplant into the animal's brain and have more and then have more awareness like a human does? Human-like external features, as you see in the picture here. Just having, again, the same thing with human stem cells being transplanted into animal embryos. Will they come out human-like? Will they have external features of humans? And again, the same concept with human-like gametes. So the stem cells being transplanted into the sperm or the ovum of the embryo. So will the animal eventually, if the animal eventually mates with another species, will that fetus be human-like? So these are the ethical concerns that are regarding the human animal transplant.
Okay, from a faith integration part, from the theological and ethical point of view, the therapeutic intent of xenotransplantation can be appreciated. Despite the religious notion of a man's significant uniqueness in creation, animals are acknowledged as fellow creatures as well. However, not everybody would agree with this type of care, so personal religious beliefs or reasons should be respected. And the most fundamental conclusion is that, according to the perspectives of Christianity, there are no specifically religious, fundamental, and generally binding reasons to prohibit this type of transplantations as a means of treating grave and life-threatening organ insufficiencies, as long as these animals are treated with the appropriate attentiveness and respectability and responsibility, of course.
These are references. And concludes our presentation. Let's see.